Thank you for the wonderful introductions. Are there any questions? Rosemary and I like to thank your hospitality. We are honored to be here with you. It's been a great day. It's been a bit of a long day, but there were fantastic presentations all the way through, including the previous session. So I'm George, and I'm from Berkeley, and thank you, Inigo, for your hospitality also. So, in conclusion, seriously, it's late in the day, and there are a lot of full professors here, so they need this. There's glycolytic metabolism. There's oxidative metabolism. The product of one is the substrate for the other. And the link between the two is lactate. So we have this exchange of energy in form of carbon. And this exchange can occur in a cell. It can occur between cells in the tissue, between organs in the body. So we now know that lactate is actually, and I will show you, a preferred fuel over glucose. It's the major gluconeogenic precursor. It's a signaling molecule. And our most recent work indicates that this shuttling of carbon occurs not just during exercise, but it happens all the time, including and especially when we eat. So 50 years ago when I started, well, I actually started more than 50 years ago, lactate was persona non grata. It was an indicator of oxygen lack, a waste product, a fatigue agent, in fact, a poison. Where do the ideas come from? So now I'm going to take you on a journey, and please come with me on this journey. It's really the course of my scientific career. So the history of interest by me was as an 18-year-old person running 440 yards in a place called Madison Square Garden. I show this picture to my students and sometimes even my children. They don't recognize it's me. But everybody was 18 at some time. So, but I didn't achieve like I wanted. So I asked my coach, Coach Patty, why can't I make the Olympic team? He said, you have too much lactic acid. You have an oxygen debt. Oh my gosh, first I had original sin, now I have lactic acidosis, and I have an oxygen debt. What is it? Why do I have so much? He said, go read about it. So I read about this man that Inigo mentioned, Otto Meyerhoff, Nobel laureate. He had a half of a frog. And I'm trying to use the cursor here. I know that our previous speakers have tried to use the pointer, it doesn't work. But you can see there's a half a frog in this bottle. Now, sometimes people ask me, what happened to the other half of the frog? I don't know. But you can see that there's no oxygen. There's no perfusion. There are there's electrical stimulation. These muscles contract and contract until they stop. And then when they stopped, there was a lot of lactate. And there was a lot of acid. But when we think about this now, we realize that when the muscle is made to contract, it needs to generate ATP. And if it doesn't have oxygen, it has to glycolyze. And so this, in the history of chemistry, was really important. So a certain amount of glycogen was broken down and a certain amount of lactate appeared. So this was the quantitative calibration of glycolysis. This was very important, but physiologists took these results to imply that the fatigue was caused by the lactate, that lactic acidosis was a bad thing. that there was subsequently an anaerobic threshold. And this substance, instead of being not only benign but helpful, was actually a poison and involved in conditions such as sepsis. So it was all bad news reading for me. And furthermore, the professor of Meyerhoff was a man named Otto Warburg, who's here pictured studying cancer cells. And if you know a little German, wenig means very little, and viel means a lot. But regardless of the presence or absence of oxygen in cancer cells, break down glucose very fast and make lactate under fully aerobic conditions. This was thought to be unique to cancer, but in fact, it's typical. Like, it's happening right now in us all. So anyhow, young man's dilemma: lactic acidosis, fatigues, cramp, oxygen debt, maybe cancer. This is how I set out to become a scientist. So now we know that lactate is, the idea is different. It's actually a fuel source, and I'll show you this. Preferred over glucose. It's the major gluconeogenic precursor. It's a sizzling molecule. We call it a lacthormone, but people don't like that word. Neither do we, actually. But it is a major means also, not only of delivering energy during exercise or exchanging exercise, but after also eating carbohydrate. So, and there actually is application to treatment of injuries and illnesses. So here's the old view of glycolysis. Glucose breaks down to triose phosphates, pyruvate is formed, and then under aerobic conditions, that is with oxygen, the pyruvate goes into the mitochondria. And when there is no oxygen or inadequate oxygen, lactate is made. I submit this is wrong. Rather, this is what a contemporary view is. Glucose breaks down to the triose phosphates. Pyruvate is formed. And I guess maybe I can get the cursor to work. But most of it is converted to lactate. That's fine because the lactate enters the mitochondria. So does pyruvate. But right now in you, maybe me a little bit more, you have at least 10 times more lactate than pyruvate. And what's going into your mitochondria when you exercise, and the ratio is now not 10 but 500 or almost close to 1,000 sometimes, what is going to get metabolized is lactate. So here's the original idea of the lactate shuttle hypothesis. This was work based on our work and those of Ken Baldwin and Peter Harchachka, that when a fast glycolytic muscle contracts, glycogen is degraded to lactate, and this lactate will enter the circulation and go around the body and raise the blood lactate level. But very quickly during exercise, that blood reperfuses an oxidative fiber, and together with the lactate from the adjacent glycolytic fiber, enters the mitochondria and is burned. So we see here what's the idea, the hypothesis is that there's simultaneous production of lactate and there's simultaneous consumption of lactate and disposal mainly by oxygen. Well, subsequently, we realized that we didn't think about the brain. We were exercise physiologists, but this happens also in the brain and to the brain. And we didn't think much about the liver, but the liver is important. We didn't think about signaling, but that will arise as we go through this talk. And transport is implied, but the data were missing. So the key concepts from this is that there are producer cells or tissues that give rise to lactate, and we call these driver cells because they're driving the flux of carbon. And there are recipient cells that use this. And not only can tissues become drivers, but they can switch to consumers, and this is true of muscle. And what the most important part of this might be the presence of the mitochondrial reticulum or network. Because in biology, like in physical world, things move from high pressure to low pressure, from high concentration to low concentration. And mitochondria or the mitochondrial network is important because it is the sink. It draws this energy flow. So we're going to talk a bit about mitochondria. So here is one of the original setups. This is a rat, and it's in a chamber that we made out of plexiglass, and here's a treadmill which was built for us. This is Dr. Casey Donovan at the University of Southern California, and this is somebody else in the room right now. And my wife likes this picture because I had brown hair. Well, that was fine, and we've got quite a bit of data, and it was on rats, and we show it that the title of the paper was Training Increases Lactate Clearance, not change production. We know that after you are trained, your lactate's lower, but we found in the rats the production was the same, but the removal had improved, mostly by oxidation. That's where the mitochondria come in. So, okay. So then we had this subject. It was in an earlier picture. And this is the middle 80s, and so the advent of stable, non-radioactive tracers, carbon-13 labeled lactate. So we took this subject, and as you can see, we infused some labeled lactate over here. We took some blood samples over there. We collected the breath. We did rest and easy and hard exercise. Easy exercise, 50% of max. Harder exercise, 75% of VO2 max. And we did this. It seemed to work okay. We got 5 more subjects and published a paper. That is Robert Mazzio, top right-hand corner, and my friend Tom Buttinger. So actually, we didn't have a human use protocol, but Tom was head of the IRB, and he was the attending physician, so we did this experiment the first time. And subsequently, we got IRB approval. So what we can see here is that at rest, there's lactate production, quite a bit, but half of it's removed by oxidation. So there's continuous lactate production at rest and also during exercise, but most of it's removed by oxidation. And then at 50% of VO2 max, there's an increase in production of lactate, and now the relative oxidation increases to 80%. And then at 75% of VO2 max, the oxidative disposal hangs in there about 75%. So during exercise, in graded exercise, these are each separate experiments, we can see that there's lactate production and removal by oxidation. So what's happening here? A couple of key points, and I'll put up these key point slides for us because it's important to do that. So lactate production occurs continuously, and these are in post-absorptive people who hadn't eaten. And it's the obligatory product of glycolysis. If you really look at the energetics, glycolysis basically cannot stop at pyruvate because the energetics favor lactate production. But lactate is disposed of continuously. Well, who knew? Until we used a tracer, nobody could know. And we know that lactate production increases during exercise. That's not novel. But during exercise, that's submaximal, up to 75% of VO2 max, lactate's removed basically by oxidation. Who knew? Well, without a tracer, nobody knew. So then we're going to address the question, well, where in the body is all this happening? We assume that, well, during exercise, the lactate's being produced in the muscle. So we took our subject, and he went to the cardiac cath lab at UC San Francisco, and we put a number of catheters in to do these experiments. So in the left arm over here, which is on the right, we have an arterial catheter. Then over here on the right side, we have a number of venous lines. So one was inserted just above the wrist and went into the biceps, in the middle of the biceps, and that was going to represent venous drainage from an inactive muscle. And one of the catheters was advanced through a large vein up through the inferior vena cava, up through the superior vena cava, through the atrium, down the inferior vena cava into the hepatic vein, into the liver. And another one was inserted up the arm, through the heart, down into the iliac vein. So now we could sample. And finally, because this is a cardiology conference, one line was put into the coronary sinus. So if you recall, the coronary sinus is where the nutritive blood supply that went through the myocardium re-enters the circulation. It's really close to the AV node. I remember looking at my EKG and trying to put that catheter in, bumping the node. It was really a bizarre EKG. But that turned out to be without incident. But the venous line that went into the liver got wedged because the guts were moving around during exercise. And a resident in his haste decided to clear the line and push saline into my liver, and then he made some comment about, a little pate. Okay, so here are results. That hurt, by the way. So before, others have shown that fatty acids are the major fuel for the heart. That's not new. There's always an obligatory glucose supply, and about 13% in the resting person the fuel was lactate. But then even exercise at 40% of VO2 max, lactate is more than half the fuel for the working heart. And this lactate, of course, came from the muscle and probably also the skin, because there was some sympathetic stimulation. But fatty acids, even though the amount increases, relatively it's less, and there's 4% glucose. So this was the PhD dissertation of Bill Stanley. Okay, so isn't this a lactate shuttle? The muscles are fueling the heart. So during exercise, blood lactate from working muscles and other sites fuels the heart. Who knew? Without all these catheters, we wouldn't know. But think about application. Think about cardioplegia. The lactate is the major fuel for the heart. When somebody has heart failure or they have an MI, what do you give? Glucose? Or we give lactate? Give the tissue what it prefers. Okay, the next step in our journey was to determine what are the effects of training. Exercise training. Exercise is a modality to really improve body performance in the youth and especially in aging. So we had a group of men. We studied them before and after training, and we studied them in easy exercise, 45% of VO2 max, and harder exercise, 65% of VO2 max. We had femoral, arterial, and venous catheterization. We measured limb blood flow, that's leg blood flow, by the thermal dilution, and cardiac output by green dye method. We used 2 isotope tracers, carbon-13 labeled lactate. Now, most of our carbon is mass 12. So carbon-13 is heavy by 1 atomic unit. But in a mass spectrometer, we can distinguish between carbon-12 and carbon-13. And also we gave glucose, because we could study glucose and lactate simultaneously, and we used dideuteroglucose. So this glucose is heavy by mass plus 2 because deuterium has a mass of 2 instead of 1. But the trick about this is that when carbon recirculates the glycogen, it labels the glucose, and now it'll be mass plus 1 glucose. We took biopsies. We had 2 months of supervised training, 6 days a week, and these people were studied post-absorptive. So here's a video, and I'm glad the video is playing. So those white tubes are indwelling catheters in the femoral artery and vein. So on your right is skilled hands of Dr. Gene Wolfel, who is head of cardiology at the University of Colorado, and on the left is Dr. Ann Friedlander from Stanford University, and they're taking respectively arterial and venous samples, and you can see there's a lot of teamwork here. It takes a lot of people to hand the syringes, take them, and decant them and use them to measure hormones and metabolites and isotope dilution. Okay, so 2 tracers, femoral arterials and venous sampling simultaneously, measurements of carbon-13 CO2 in the venous effluent and also in the breath, and measures of flow distribution. A lot of papers published with first author Dr. Brian Bergman, who's now at the University of Colorado, and there's Gene and Ann on your right-hand side. A lot of data to show, but what I want to do here is indicate that, again, as in rats, so now, not in rats because people who didn't want to believe, well, it's just rats, doesn't matter. Well, now it's the same result is true in people. We can see after training, there's an increase in lactate clearance. There isn't much of a decrease in production. In fact, in harder exercise, there's more lactate production after training, but it's cleared, it's removed. So if we have a molecule of carbon-13 lactate going into a working muscle, that 13C comes out on the venous side as CO2. It's taken up and it's burned. So, a couple of punchlines. Here's Brian and Otto. I don't think they ever met. During exercise, the working leg simultaneously produces and consumes and oxidized lactate because we have these compartments that are drivers and other ones which are recipients. And so training improves muscle lactate oxidation. Well, who knew? Now, without the tracers and the catheters, we could see it happening in real time. So what about gluconeogenesis? Again, I indicated earlier, this carbon-13 has a signature of mass plus 1, and when that carbon goes back into a glucose that's being made during the exercise or at rest, or before or after training, then the resulting glucose is heavier by mass plus 1. So what we have here is subjects at rest, before training, and after training. And you can see this big increase in gluconeogenesis. So we know that trained people can regulate their blood sugar more easily. Well, this is why, because of gluconeogenesis goes way up with training. And then what about exercise? Well, this is the people at 45% of VO2 max before training. And you can see gluconeogenesis went up, but at 65%, gluconeogenesis is suppressed. Probably there's this shunting mechanism where less blood goes to the gut when you're not trained, so because we have to support the working muscle and the brain. But then after training, you can see this is the same power output. This was 65% max. Now it's about 54% of VO2 max, the same power output. But what we can see is that the gluconeogenesis is way up. And at 65% of VO2 max after training, you can see there's still an increase, although probably there is some shunting going on. So this explains about 25% of the lactate disposal. And again, 75 or 80% of the lactate was oxidized. And of this glucose that's going to be made from lactate, well, it's largely going to be burned too. So we can see that in terms of delivering carbohydrate energy for oxidation, lactate plays a major role. And training increases gluconeogenesis, not just during exercise, but at rest also. So a couple more punchlines here. Training improves the ability to maintain glycemia by improving the capacity for gluconeogenesis from lactate. Who knew? So when we took biochemistry or physiology, we learned about the Cori cycle, and we could say gluconeogenesis really is an expression of the Cori cycle. But who knew? Actually, not the Cories. The Cories never did a human study. They never did experiments on humans, either before or after training. So, what about this lactate stuff? Can you give it? Can you eat it? Well, anybody ever had sauerkraut or kimchi or sourdough bread or maybe yogurt? Yeah, you can eat it. Can you drink it? Can you put it in a sports drink? Yeah, we did that. Can you use it to improve performance? Can you use it as a treatment for illnesses and injuries? Yes. So here's a couple of references by Dr. Benjamin Miller, who's now at the University of Colorado Health Science Center in Oklahoma. I'm having trouble between red states and blue states. Please forgive me. Anyhow, Ben is there now in Oklahoma and doing very well. But we developed this technique where we basically infuse sodium lactate to an arterial level of 4 millimolar, which is considered to be an exercise, pretty hard exercise in some cases. And when we do that, we had our carbon labeled lactate, and we had our measurements of glucose metabolism with deuterium. When we gave lactate exogenously, it suppressed glucose use. So lactate is substituting for glucose, and there's being the salt of an acid, lactate and ion is actually a base. So we had small increases in blood pH. So thinking about managing acidosis by giving the salt of an acid that's organic and appropriate for physiology, think about managing acidosis by infusing lactate. Now, I think this is our most impressive result so far in terms of showing the superiority of lactate over glucose as an energy substrate. This was in collaboration with UCLA neuroscience, neurosurgery. And on your left, you can see the unfortunate case if somebody has a left frontal lobe injury. So this is a positron emission tomograph picture when labeled glucose was given, deoxyglucose, which is consumed but not metabolized. This is used also in cancer research. So these so-called red hotspots indicate where there's high metabolism, in this case of glucose. And you can see here where the injury is. Here, the left frontal lobe. On the right side, there's good metabolism. Here, it's blocked. Next day, we infused lactate. It completely suppressed glucose uptake. Lactate preferred over glucose, not just in the muscle, but especially in the brain. So now think about lactate infusion as a treatment for traumatic brain injury. Okay, we're going to switch gears now. So I've been talking about carbon fluxes in between tissues, but I did advance the idea that we have to have this metabolic sink. This material has to go somewhere, and someplace that's oxidizable, the mitochondrial network. Now, I tried not to say mitochondria, Because if you look at this, you can see that we don't have these little spheres or blobs. We have a whole network that's interconnected, that goes from the cell surface deep within here, a muscle fiber. I've chosen here not to show our early electron micrographs on this, but I prefer to show one by Hans Hoppler from Bern, Switzerland, who has shown this on human skeletal muscle. So if you do a section through here, you'll see a sphere, or there you see a sphere. Wherever you cut through here, take a picture, you'll see a sphere. But if you, right along the Z line, you get the expression of the mitochondrial reticulum, or network. Here's our one of our most recent studies. This is on a mouse muscle. So we're using immunocytochemistry here. And what you can do is you can have an antibody to, for instance here, green, which is the lactate transporter, MCT1. And so where you see the green, what's happened here is a laser has hit this protein complex that's been labeled with a fluor made to fluoresce green with a certain wavelength of light. And look at the abundance of the green around this muscle fiber. These are the cell membrane lactate transporters. Now also, on the inside, you can't see it because it's co-localized. We had another antibody, and it attached itself to the mitochondrial pyruvate carrier. So if we remember our primary colors, when we mix green and red, what color do we get? You athletes know this. You want to go for the, come on. You want to go for the gold. So where you have co-localization of the pyruvate and lactate transporters in the mitochondria, you see the mitochondrial network. So this MCT lactate transporter stuff is just not on the cell surface, it's deep in the cell and it's in the mitochondrial network. This is under review for publication. But you can tell anybody about it, because I think it's really kind of cool. Okay, now we're in the home stretch, folks. I know it's late. Your patience for a few more minutes. We were asked to write about a review by the Journal of Physiology about lactate in contemporary biology, and we decided to call it the Phoenix Risen. So if you look across the bottom here, we have this hellacious place attributable to lactic acid, burn, cramps, oxygen debt, anaerobic threshold. This is the fires. And then the phoenix, we now know that lactate has all these positive effects. Cell signaling, fueling, it's involved in adaptation because it stimulates gene expression. So this is sort of the idea right now. We have this phoenix rising, a completely different attitude about lactate. And one of the things in the article that fortunately there was a really good artist for the Journal of Physiology, and she drew a cartoon of the lactate shuttle for somebody exercising. And down here, and of course this is way out of scale, here's the working muscle and the skin releasing lactate into the circulation, and it's going to the liver and the heart and the brain. But also somebody taking carbohydrate, like a drink or maybe some pasta. And so the metabolism is starting in the gut. That was the hypothesis. So we're calling this now the postprandial lactate shuttle. So this was a hypothesis. And there was evidence for the hypothesis by others on rats and dogs. If you give dietary glucose and it's absorbed, it circulates to muscle, and muscle, even especially red muscle, which is highly perfused, gets most of this glucose, And so there's a rise in lactate from glucose that reaches muscle. There's a dietary glucose that circulates and gets to the muscle. This has been seen before, and it's sometimes called the indirect pathway. I don't know if you heard that term, the indirect pathway, and there's quite a few publications on it. But also others have put hepatic portal catheters into rats and then given them glucose by gavage and looked to see what appeared in the hepatic portal vein. This is the vein between the GI tract and the liver. And you could see the first thing to appear in the portal vein after giving glucose is not glucose, but it's lactate. But there were no data on humans, so we did this study. We had over 36 subjects. That's over 35, even at this time of day. And so we have our tracers over here coming in through a pump, and the tracers are of glucose and lactate and glycerol. Glycerol is a product of lipolysis, so we're trying to get at not the glucose lactate carbohydrate interactions, but also get a measure of the lipolysis which is going on. And so that's going into a forearm vein in this side of the body. And then, and this is after, this is at rest. This is not during exercise. We did do exercise on these subjects to compare fit men and unfit men, young men and old men, and same thing for women. And so then on the other side over here, we have an arterialized sampling site, and we're measuring the concentrations and isotopic enrichments of glucose, lactate, glycerol, and fatty acids. And also we have insulin, we're measuring insulin and the insulin antagonists. And also, importantly, with our metabolic cart, we're measuring the gas exchange values, VO2, VCO2, and also the V13 CO2. So we're getting oxidation. Okay, this is really a key result right here. When you have people studied, and I didn't mention it, to set this up, we got to the lab at 4:30. So myself, a nurse, 4 graduate students, and 10 undergraduates to make this study go. And nobody complained. So, and we had a 90-minute equilibration period for the tracers. Here's the arterial lactate. It's slow to begin with, is falling because people are getting pretty hungry right now. And then right at 7 a.m., the supper they had was finished at 7 p.m. They took 75 grams of glucose. This is an oral glucose tolerance test. And you can see what happens here. Now, this is lactate after a glucose challenge. There's a small but significant increase in 5 minutes. So again, ladies and gentlemen, we gave glucose. The first thing to appear in the blood is lactate. And it rises a peak in about 15 minutes, and then it starts to decline significantly, and then it rises again over the course of time. So we see there's something that's happening right away, probably in the gut. So we call this the enteric phase. And then the other part is happening subsequently, and we call this the systemic phase. So arterial lactate rises in 2 phases following an oral glucose tolerance test. And again, we had tracer measurements to know that this rise was due to production. What about glucose? Well, it's important to compare lactate and glucose. So here we just have, we're taking things from right when people took the glucose. There's no significant increase in 5 minutes. But there is at 15 and subsequently. And so the traditional glucose tolerance curve, when people start measuring after 30 minutes or after an hour, yeah, we replicate that, but there's no significant increase in the first 5 minutes. So the first thing into the blood with a glucose challenge is not glucose, but lactate. And compared to lactate kinetics, the glucose response is slow and it's delayed. With our methods now, we need to add up how much of the glucose was disposed of by this mechanism. So in the yellow, we have the amount of glucose that was consumed that appeared as lactate. And then in green, we have the glucose that was formed from lactate. And so there was about 50% of the oral glucose load was accounted for directly or indirectly as lactate. Okay, so I started off by saying or suggesting that lactate shuttling is involved in energy substrate delivery. But we also know there's a crossover concept in which different substrates compete against each other. So if we use more carbohydrate, we're going to use less fat, and vice versa. If we're going to use more lactate, we're going to use less glucose. So there's this ongoing, this concept of ongoing substrate competition. So we now know that there's fuel, cell fueling, there's gluconeogenesis, there's cell-cell signaling, there's satiety after eating. Those of you who are athletes and go for 400-meter run all out, or 1,500-meter run, or the swimming equivalent, or cycling equivalent, you get through, you are not hungry, because the lactate's crossing the blood-brain barrier and suppressing our hunger centers in our brain. And one other thing we're doing, we're finding that there's multiple signaling functions of lactate, including blastocyst development. So we actually have a grant with the NIH to study the role of lactate in blastocyst development. It's essential for that. So thank you for your attention. This is the second study. I was the first subject. Dr. Agostini was the second subject. And this is the day we studied rosemary. That's me, that's her. These are some of our undergraduates and graduate students. And there are about 12 people in the room. And here's, I can't say Joshua's name, but he was the local college shot put champion, a national champion, All-America athlete. He participated. So we have good gender diversity in our lab, and we have good ethnic diversity as well. So this is where we started. Think about the fact that there's glycolysis. There are cells and tissues that produce lactate. We call these drivers. And then this stuff has to be disposed of, and it's disposed of mostly by oxidation. And so we have this lactate shuttle, because lactate is the intermediary. It's the product of glycolysis, and it's the oxidative substrate. It crowds out pyruvate from the mitochondria, so it's the major oxidative fuel. And now we know that lactate is a major route of carbon delivery, not just exercise, but after carbohydrate nutrition. Thank you for your attention. We are open for discussion. It was a short trip, but here we are. Are we going to have questions? Push the button on the right.
Sí. ¿Preguntas? ¿Tiene alguien alguna pregunta? Ahí hay una pregunta. Mario.
So, George, thank you, first of all. I want to ask you if there is any difference between the level of lactate in capillary or venous or arterial. Is there any difference? Because sometimes with Javi, when we measure in our courses or with our patients, we have a really high levels of lactate in rest. But then when they do the stress test, they get a really good performance. So I don't know if this is normal, it's not. I have many questions about this.
Okay, well, you actually asked me several questions, but
Yes.
they're all
It's
they're all
not
really,
one.
really good. So lactate's high. So when I first presented this, it was at the American Diabetes meeting, and there was a physician there from the NIH, National Institute of Health, and they're doing carbohydrate feeding studies to healthy people, and they get blood lactates of 2 millimolar, so that they think that carbohydrate is causing sepsis. No, it's the way the body works. Okay, so if lactate's high, maybe because you're testing somebody and have a sympathetic response.
That's my question.
Yeah.
Yeah, because
Yeah,
I
yeah,
don't know
but-
if stress or
Stress.
Other things can change the levels of lactate, but really high, like 3.6, 4.4.
Yeah. Well, you can do that with epinephrine.
Great.
Yeah, and actually, we downloaded the short version of the talk. The longer version of the talk has that plot right after the sympathetic effect on lactate level.
And is there any protocol that we can use to make sure that this goes down?
Yeah, so I think people who do clinical studies here, you'll get an anxious participant subject. And you can tell that by their breathing, and if you're going to measure their lactate, you can tell by their respiratory gas exchange. So what you do is you wait a little while, right?
Okay.
And you let things calm down. The turnover of epinephrine is really very fast. The half-life is 2.5 minutes. So even if it's high, if you just wait a while, the lactate will come down.
Thank you.
First of all, George, thank you for your presentation. It's definitely a very, very good presentation. We are cardiologists. A lot of people here are cardiologists. Always have thought about lactate as a bad person of the film. I mean, the cardiogenic shock in a very serious situation. Only with lactate, about 5, you can be in a very, very vital situation, very, very serious.
Mm-hmm. Yeah.
We have to adaptate our thoughts, not thinking that it's a bad person of the film, but one very useful fuel for the body in that critical situation. And the other questions, the glucose infusions that we have used in several trials for the myocardial infarction, the trials have been neutral. Do you think that another kind of perfusion like Ringer lactate could be better as treatment for this situation?
Yeah, so thank you for the questions, but I think you actually answered the first part about our thinking. So we think of lactate as a stress, but it's actually a strain, pushback. So you have an injured heart, it needs energy. It should glycolyze to support ATP levels. So you should see lactate high. So lactate is a good biomarker for stress, because what the body's doing against this stress, it has a strain. So stress and strain. So I tell this little story that Rosemary likes me to tell. Okay, so you and I are related by some reason, 23andMe. And it turns out we also have a cousin who lives in Samoa, in the jungle. Okay, so this is kind of wild, but So we asked him to come visit us, and we're at San Francisco Airport called SFO. And he's been living in the jungle, and fire is really a commodity. It's hard to have a fire or cook something in the jungle. Everything's wet all the time. So we pick him up and we welcome him to the family and all of that. And then we're driving along. There's a big fire. And there's the fire trucks and the firemen are running all around. And he says, those guys really know how to make a fire. But and that's what happened historically. Lactate appears when there's stress. But if it's strain, it's the body's working against it. Now, to your second question, you asked about this glucose-lactate competition. We see it clearly in the brain. So after traumatic brain injury, glucose uptake goes down almost 50%. Probably there's a redox change at GAP-DH. So our colleagues, neurosurgeons and emergency physicians, try to fuel the brain by giving glucose, D5 glucose, raising the glucose level into the diabetic range. But glycolysis is blocked, so that does nothing. So then they'll try to give insulin, but the brain doesn't have GLUT4, which is insulin responsive, as GLUT1, which is concentration responsive, but even that doesn't work. But we've shown that we can restore the carbohydrate energy flux by giving lactate. So there is this competition. If you partially block one arm, like glycolysis, then you can substitute with lactate.
Well, in the first place, thank you very much, Dr. Brooks. I must say that you have huge fans here in Spain, and I never thought I'd be witnessing a talk of yours. So thank you very much for your presence here and for your talk. My question is more about performance than of health. You started the talk by saying that your coach had said that you had too much lactic acid in your blood during competitions. With what you know today, do you think that's true or not? And if not, what was the limitation to performance in your case in your experience as a scientist, not only as an athlete?
Yeah, I could have trained more to improve my clearance.
And what do you think you should have done to improve that?
Well, I should have segmented my training into phases and done a lot of aerobic training early in the year. And we know that, and actually we have the master here who can explain this. Zone 2 training develops the mitochondrial network. So that you build the base and then you work from there. So, but you know, these races, they were right after, they're in January basically. So it's really early in the track season. So we weren't trained as much as we needed to be. And then in New York City, summer didn't come until after school was out. So then we had our best times running for the clubs in the summertime. Zone 2. Thank you. Thank you for the compliment and the question. It's hard to see up here, the lights in our eyes. I think, is it cerveza
¿Una
time?
última pregunta o nos vamos a tomar una cerveza? So, muchísimas gracias. Thank you very much, Dr. George Brooks, for your great presentation.